Good afternoon, everyone. I'm delighted to be able to take part in this HELP seminar, ethics seminar for biology. And I'm delighted that you have spared some time to be a part of this very important session. And I'm Crescent Musime. I'm going to be sharing with us on some things concerning biology, A-level biology, particularly the theory, in the next almost 2 hours, because we have taken some of that time. So I pray that the network will be good and we'll be able to have a very engaging discussion. Thank you. So I decided to first give some tips concerning the paper, because I know most of us, if not all of us, that are participating are sitting this paper at national level, will be sitting at national level for the first time. But I just want to confirm what the teachers have been saying to you concerning how the paper is organized. First of all, our paper is 530/1, that is paper 1, or 530/2, that is paper 2. So in paper 1, as you already know, we have multiple choice questions, 40 of them, and then 6 structured questions, all to be done in 2.5 hours. The same applies to paper 2. Now, the challenge that candidates usually encounter is failure to finish, and this can be attributed to the fact that they don't plan for the time very well. So if you're given 2.5 hours and you want to earn 100 marks, so it's very important that for each mark you're going to earn, you know how much time it will take you. So I usually advise the candidates to look at the marks available and the time available. So when we look at that, we'll notice that you earn 1 mark in 1.5 minutes. So that means that if you're doing paper 1 and you're looking at section A, those are 40 marks times 1.5 minutes. We should give you 1 hour. So it means that you should never exceed 1 hour when you're attempting section A. And so that means the 1.5 hours are left over for section B. So it means that you can be able to track whether you're taking too much time on one number or you've taken extremely low time. So if we are looking at paper 1, for instance, in section B, it means that each number should not be done in more than 15 minutes. So if you find yourself spending 20 minutes on one number, it means that you're cheating time for another number. However, if you take 5 minutes, then that is also something you need to be concerned about. So it means that the time that has been allocated is more than enough for you to plan your answer and to attempt the question thoroughly. So it's very important that you understand the question before you even attempt it. And after that, then you plan the answering. Because what usually happens in paper 1 is that the space left is not enough for some candidates. And what causes this is because they don't plan their answer. So they answer as they are thinking. Yet it is better to think first, sketch the answers somewhere, maybe scribble down the main points in the answer, then you transplant them on the paper afterwards. So that practice will help you budget for the time accordingly. Because if you write outside the space that has been provided, then it means that that work will not be marked. So it's very important to plan accordingly. Actually, paper 1 helps us to be more precise in our presentation. It helps you to be very precise because the spaces that are allocated are very, but they are budgeted for. Unlike in paper 2, where you have the luxury of a lot of space for you to write your response. But that doesn't mean that you should spend a whole page trying to earn 2 marks or 3 marks. So that brings me to the second point. The number of marks allocated for a question should guide you to know to what extent you should attempt the question. So when you are attempting that question, it's important that you keep checking yourself and asking yourself, how will this answer be marked? Where are the marking points? Because if there are 2 marks and you are writing things that cannot bring out those marks, then it means that you're not going to earn the marks. So usually with biology, if there are 2 marks...
Teacher present?
Yes, please.
Yes. Is there a screen that is having that information you're reading, or?
No, I don't have this. This one is off-head.
Okay, fine, because I think people thought
that one.
Kindly
I thought I should talk about this before I get to the actual things. I hope I didn't lose anyone in that process.
Not really, not really.
Okay, then that's great. So, where was I? So I was saying that when you look at the marks, in the past, they used not to put marks on the question, and it caused problems. So the good thing is that nowadays there are marks put at the end of each question. So that helps you to know how you will engage. So if they have 2 marks, it means that they are going to be either 2 marking points or 4 marking points. What do I mean? If there are 2 marking points, it means each one will score 1 mark. But most often than not, there are 4 marking points where each one of them will score half a mark. So that should be a guiding principle when you are attempting that question, so that you don't write too much or write too little and end up wondering why you're not scoring the marks. And so, of course, very important to identify what topic the question has been set from. By now, you should know the total number of topics in the biology syllabus. And then as you're preparing, you prepare well and know that for each of the topics you have read. It's very dangerous to spot the examiner, because most likely you'll be outspotted, because the examiner tries to balance the way they set the exam, the setter. So if you choose out your favorite topics or your easier topics, then you put yourself at a disadvantage even before you start answering the paper. So it's very important to read widely. And then thirdly or fourthly, it's important to understand how certain topics are interconnected. With biology, there's a lot of interconnectivity. And this interconnectivity must be reflected as you're answering certain questions where that interconnectivity comes in. Now, I have certain things I have prepared here concerning the way the questions are usually structured. We have these command words that I usually put on every question. I'm sure you have seen them if you're very observant. So we have the command of define. So when you're told to define, it means that you must provide something that is very precise or very clear. Okay, so if you're asked to define a concept, your answer must be clear and to the point. It should not be too wordy, because definitions are usually very precise. So it's very important that you avoid that. Then we also have another one, describe. This one is very, very common in biology. Now here you're required to give details concerning that particular concept that is being asked of you. Okay, if they ask you maybe to describe the process of cyclic photophosphorylation, it means that you're going to go into that details, the step-by-step process of cyclic photophosphorylation. So it means you're not going to outline, you're not going to give something that is brief. It needs to be comprehensive. You don't leave any stone unturned. Okay. And usually describing is mostly about the how things happen, the what is happening and the how it is happening, and possibly where it is happening. That is what description is all about. But then we also have another one, which is explain, which is also very common in biology. Now here, you don't only describe what is taking place or what happens or what would happen, but you also go ahead and give reasons. So you're talking about the how, the what, the where, but even the why, and also the when may also be put there. So here you're going to try to make connections, okay, between different concepts in order to bring, to explain your point, in order for for the concept to be well understood. So it's very, very important that if there are any topics that are linked with what you're trying to explain, make sure we can see that. But then again, be guided by the marks that have been put so that you don't go overboard and write more than is required or more than can be scored for that question. Then compare is also another very common question. Here you're going to look at similarities and the differences. So between two concepts, usually the concepts are somewhat related or somewhat, they're in the same topic, but they need you to show how they are similar and in what ways they may be different. And it's very important that you're able to, for instance, when you're talking about, when you're comparing, you need to have a basis of comparison, a criterion for comparing. In some of the questions I have prepared, I'll show you what I mean by a criterion for comparison. So if you're comparing, for instance, let me just give this example. If you're comparing cyclic photophosphorylation and non-cyclic, what aspect of the two are you comparing? Is it the types of photosystems involved? Is it the movement of the electrons? Is it the products? So at the back of your mind, ask yourself, what is it that I am comparing? Okay, so when we are comparing, we usually have similarities and differences. In the past, they would ask questions where they would say compare and contrast. Now, in such a case, it means they clearly want you to write similarities and differences. But nowadays, they just simply ask you to compare. So when they ask you to compare, it means that they want similarities and differences, not just the differences between the concepts that are being examined. Then we have discuss. This one, not to, you don't come out openly like they say discuss, but when they write the explanation, when they ask you to explain, usually there's an aspect of discussion involved. So here they want you to provide a detailed analysis of that topic or that question that has been given to you. Okay, and here there must be arguments and viewpoints, an evaluation of some kind. Okay, for instance, they tell you that life without water would be impossible. Discuss. So you would now need to look at what water is and how important it is. Okay, so it's very important that you're able to spell it out very clearly. Okay, or transpiration is a necessary evil. Discuss or explain. So explain and discuss sometimes come off as the same, because even when you're discussing, you are giving a detailed answer. So the ones I have put in red are the ones that are very common in the examinations. Then we also have evaluate, not that common, but sometimes they may ask you to evaluate. So to evaluate is to give an assessment or to judge something and to see its strengths and weaknesses. For instance, maybe there is a method that has been used to arrive at certain results, and they want you to talk about whether it is an accurate way of measuring or it has errors in it, and what are the sources of those errors, or what would be a better way of conducting a similar experiment and minimize the errors. So the evaluation here will help you when you are attempting such a question. And then we have outline. It's also very common. Here they want you to just give a brief overview or summary of the main features, okay? And they must be in a structured format. So if they ask you to outline the stages of meiosis or the stages of mitosis, so it means that you're going to look at the key features. What are the different stages within mitosis, and what is the main thing that happens in each of those stages? Okay, so you don't go into so much detail. And these ones are typical of section B, paper 1, and a bit sometimes in paper 2. Okay, then summarize is more like this other thing above. It's very rare they'll tell you outwardly that you summarize, but if they say outline, then it is more like the same. Illustrate also is another command word. Sometimes they may want you to annotate your diagrams, so that's where the illustrations may come in. By annotation, I mean, for instance, if you are looking at the different tissues that are found in the dicot stem, then maybe it may be important that you don't stop at writing about them, but you can also illustrate them with drawings. So when you write a few words accompanying drawings, then we say that you are annotating. So you're writing a few explanatory notes to accompany them. Analyze, that's what it is, but these are not very common for us, but they are always captured with explain. And then interpret, this one may come maybe if you have graphical work and they want you to make sense out of the graphical work. Again, this one has an aspect of the explaining it. So you'll find that the questions I put first are the ones that are the most common, but even these other words are good for us to know, because it's not cast in stone that they must always ask them the way they have always done. Maybe they may ask in another way, and if they did, then you should be prepared to answer the questions appropriately. Yeah, so these are some of the words that I thought our candidates need to know very well and be prepared to ably answer the questions when they come across them. So I prepared a few items that I want us to go through so that we can be able to see how certain questions are attempted. So I have a question that is coming from the topic of inheritance and variation and inheritance. And we are asked here to differentiate between polyploidy and aneuploidy, and then also to differentiate between linkage, and epistasis. Now, these questions I expect the participants to be able to also take part in the discussion. I don't want to make this one-way traffic. I'm sure you have covered some of these things. Then when we have discussed, then I can show you my prepared answers. I hope that will be possible for us to easily participate. So what is polyploidy and what is aneuploidy? Here they've asked us to differentiate. So we need us to differentiate what is expected of us. Maybe you first tell us the approach, then you go ahead and tell us what you consider to be the answer. Mr. Kaziba, would you mind being my monitor for today? Okay, I think. Okay, thank you. So kindly put up your hand if you have the answer to the question. Or you can put it in the chat. You can see most people are quiet. Someone seems to want me to share the screen from Chinyara SS. I just thought I'm doing just that. I don't know what could be happening from your ends. Okay, so when they ask us to differentiate, as you can see the word, it has differentiate, which comes from the word difference. So here they want us to clearly spell out the differences between the concept of polyploidy and the concept of aneuploidy. So these are concepts you must have encountered when you are dealing with mutations. Remember, mutations are about changes in the structure or amount of DNA or chromosomes in a cell. So when some changes take place to the structure of the DNA or to the amount of the DNA, then we say that mutations have happened. So some of these mutations result in polyploids, while others result in aneuploids. So this is what I have for us. Sorry. So polyploidy, when you hear the word poly, it means many. And then ploid is to do with a set of chromosomes. So here the organism will have a whole set of chromosomes added to it. So for instance, if we have, like in humans, we have 23 pairs of chromosomes. Those are 46 chromosomes.
A minute is coming back. Just be patient. It's getting back. It's getting back. He has some network issues, but Teacher Crescent is getting back. Okay. Teacher Crescent.
Mr. Kaziba. Sorry about the network issue, but I was trying to differentiate between polyploidy and aneuploidy. So we're saying that if we look at an organism that has a haploid number, say of 7, then if we look at its diploid number, it will be 2n. So sometimes mutations occur or something happens in the course of cell division that results in that number increasing. And when it increases, it's going to increase in such a way that there will be a whole set of chromosomes. So, and this polyploidy is usually as a result of some challenges or certain problems that happen during cell division. So what happens is that during cell division, the spindle fibers fail to form, and then the chromosomes fail to separate. So you find that because they are failed to separate, then the cell that is produced has extra chromosomes, okay? A whole set of extra chromosomes. So that's why you have 3n. You can even have, if it was 2n, then it can be 4n, okay? And so on and so forth. But then when we look at aneuploidy, here we are going to have addition or reduction of just one chromosome or 2 chromosomes, something like that. It means that what happens is that, for instance, if we look at Down's syndrome, in Down syndrome, we have an extra chromosome that remains, okay? So we have chromosome 21. So chromosome 21, there are 2 chromosomes that are supposed to separate at anaphase. But what happens is that those 2 chromosomes don't separate. So both of them remain in the daughter cell. And when they remain in the daughter cell, it means that one of the daughter cells will have 1 chromosome less, while the other one will have 1 chromosome more. So if that chromosome, that cell that has 1 chromosome more is, say, for instance, a sperm cell, and it fertilizes an egg which has the normal number of chromosomes that an egg should have, that is 23 chromosomes, then you're going to end up having 23 from the egg cell plus 24 got. So that's why we have what we call Down syndrome and many other kinds of situations like that. For instance, you may have Klinefelters and so on and so forth. So when you have an extra chromosome, then that's the outcome is said to be an aneuploid. Or if you have a deficiency in the number of chromosomes, then that cell is also said to be an euploid. So from what I have said here, we can see that polyploidy is caused by non-disjunction. Non-disjunction is where the spindles fail to form, okay, during prophase 1 when they should form. That means that anaphase, the chromosomes cannot separate because the spindles form in prophase 1, but in anaphase, that's when the chromosomes should now be separating, being pulled by those spindles that were formed during prophase. Then, of course, here the non-disjunction is just because the failure of homologous chromosomes to separate. Okay, so you can see that both are caused by non-disjunction, but non-disjunction of what? Here is non-disjunction of for the spindles, all the spindles to form. So it means that none of the chromosomes can actually separate for the case of polyploidy. But here for aneuploidy, some of them may separate while others may not. So you find that, for instance, one of the homologous pairs may fail to separate while all the others have separated. And that's why we have only an additional extra number of chromosomes, okay? Or a reduction in the number of chromosomes. I hope that is clear. Then polyploidy is typically seen in plants and less seen in animals. Animals that undergo polyploidy usually die because of it's not sustainable for them, because animals usually undergo, those that undergo sexual reproduction, it doesn't work out. But for plants, they can even sometimes undergo a sexual reproduction, and then that situation can be cause them to survive. Then aneuploidy happens in animals more than it happens in plants. And when it does happen in plants, these are polyploidy, we see increase in the size of the organs or the cells. We have seen these oranges that are so big, or mangoes that are so big, or tomatoes that are so big. Some of them are as a result of polyploidy that probably took place. Yes, Hussein Ismail. I can see Hussein has put up his hand. Kindly unmute and ask your question or make your contribution. Okay, I think let's carry on. Then for aneuploidy, the organisms tend to remain the same size, okay? It doesn't affect them like polyploidy affects the organisms that experience it. And because of that increase in size in plants, it usually confers certain advantages in those organisms. Sometimes those organisms will be able to produce maybe more food. For instance, they may have bigger storage organs, so they may have more storage of food, which will help them maybe in terms of drought or in adverse conditions. Or they may make them better adapted to the ever-changing environmental conditions. But then aneuploidy is usually disadvantageous. Okay, like we know people who have extra sex chromosomes, the challenges they have, or people who have trisomy 21, the challenges that they do have. So aneuploidy is usually disadvantageous. And the aneuploidy is typically naturally induced, whereas polyploidy can even be chemically induced, much as it can also happen naturally. So if you are asked to differentiate between polyploid and aneuploidy, then you need to ask yourself how many marks have been given. Then you can then answer the question. So if it is a paper 2 question, usually the marks are many, and you are free to express yourself because you have the space. But now with paper 1, it's very important that now you summarize your answers so that they can fit in the space provided, and so that you earn all the marks. So I hope that is clear. Then the other part that we were asked to differentiate was between sex linkage and epistasis. Sex linkage and epistasis. So this is another concept in genetics, and candidates usually struggle with those different concepts of inheritance. Now, I would like to encourage you to get to know all the cases of inheritance that are in agreement with Mendel's laws, and then all those that are not in agreement with Mendel's laws. Then once you have gotten them, then it's important that you try to appreciate the ratios, because with genetics, it's usually the ratios that help us understand the mode of inheritance. There are ratios, they can help us, but also when you read through the text of the question, you can be able to determine whether it is sex linkage or it is just ordinary linkage. Ordinary linkage, we mean autosomal linked genes, or whether it is epistasis, or they are lethal genes, or otherwise, or there's codominance, incomplete dominance, and so on and so forth. So now, if we are asked to talk about sex linkage and epistasis, here are the differences. Here we see that the genes are carried on the sex chromosomes, hence the word sex linkage. Now, if you don't put the word sex linkage, then we are referring to autosomal linkage, where we are looking at genes carried on chromosomes other than the sex chromosomes. So when genes are carried on one chromosome, they form what we call a linkage group. So all genes carried on one chromosome are said to be linked genes. So they form what we call a linkage group. So all genes carried on the sex chromosomes are said to be sex linked. Now, when we look at epistasis, epistasis is where we find genes that are carried on separate chromosomes, okay? They are carried on separate chromosomes, and these chromosomes are usually autosomal chromosomes, okay? So you can have chromosome number 1 and chromosome number 20, and where we are having these two genes having an interaction in the way they affect the phenotype of that organism, much as they are on different chromosomes, yet they two work together or somehow influence each other in the outcome of the phenotype. So some people may want to think they are linked, but they are not linked because they are carried on separate chromosomes. But one of the genes suppresses the expression of another gene on a totally different chromosome. So we say that gene is said to be epistatic. The word epi means on top. So it is like on top of the other one. Then the gene that is being suppressed is said to be hypostatic. So sex linkage, these are genes that are on the sex chromosome. Then epistasis, these are two different genes carried on separate chromosomes, which separate chromosomes are usually autosomal chromosomes. And when we look at sex linkage, these genes on the sex chromosomes usually determine only one characteristic, okay? They are not under the influence of another characteristic. They usually work independently, okay? But epistatic genes, these ones, they don't work independently. So you find that there could be 2, there could be 3, there could be 4 different genes interacting, but determining only one character. But for sex linkage, we have one gene determining one character. I hope that is very clear. Then, when we look at sex linkage, still, we see the presence of a gene on one gene locus does not suppress the effect of any other. I think I talked about that already. But for epistasis, the presence of a gene on one locus, remember the locus is the position where a gene is found on a chromosome or on the DNA. So you find that these genes are on totally different loci and on separate chromosomes, yet the presence of one gene suppresses the expression of the other gene. So that's why we see epistasis. Yeah, so that is for the differentiation. So like I told you earlier, it's important that you know the criteria that you're going to use when you're differentiating. So for the first criterion, we are looking at the location of those genes. Are they on sex chromosomes or are they on autosomes? Then the second criterion is how many genes are involved in sex linkage and epistasis. Then the third criterion is how these genes are expressed. Are they expressed as a result of interacting with other genes, or they are expressed independent of any other gene? Okay, so that is what I was meaning when I said that know the criterion for differentiating. If you don't know that criterion, then it will be difficult for you to avoid repeating yourself, or it will be difficult for you to remember all the different differences that are there. And it helps you also when you are revising, when you have a criteria for differentiating something. And this criteria comes from in-depth reading. When you read and you analyze, okay, you'll be able to arrive at that. And I'm sure we are all able to do that if we only exert ourselves a little more. So the second question, I hope this time I'll have people coming to participate, is still on the topic of genetics. We are told that an onion bulb has two, okay, we are considering 2 genes on the onion bulb, where they're telling us when at least both the dominant allele R and W are present in the genotype, then the color of the onion is red. Okay, notice we have 2 different colors. Okay, and then we have 2 different letters. So that should be able to start triggering your thinking in a particular direction. And then when at least a dominant allele W is present, so here it's important that we underline some of the keywords when we are writing, when we are attempting, they're saying at least. So what does at least mean? At least means that at the very least, okay? So at the very least, or at the lowest concentration, or at the lowest number. So when there is at least a dominant allele W, so it means at most you can have 2 capital W's, but at least you can have 1W, okay? So when at least the dominant allele W is present, while the other alleles are being recessive, the color of the onion is yellow. So what does this mean? That if we had maybe W like this is small, and then you have the capital, and then you have the others are small letters, something like that. So if we have such a situation, okay, small w, capital W, and then small r, then it will tend to be yellow, okay? Then when the allele small w is homozygous in the genotype, the onion is white, okay? So these three pieces of information are going to help us to answer the question. So it will help us write all the possible permutations that can arise when we are dealing with this question, okay? So give the possible genotypes for the red, yellow, and white colors of the onion. Now with genetics, this is a very interesting topic, which is very easy to pass, but also very easy to fail as long as you have failed to understand the concept. But if you understand the concept, you can score all the marks that are available. So I want to hear from the participants what they think about this question. You can see there's some group chat. There's some chat in the people are writing some things. So when I look at what's the difference between variation caused by gene recombination and mutation? Okay. I hope some of the questions don't take us far off track. But when you look at variation caused by gene recombination, that variation can be reversed, okay? It's like what happens when you have crossing over, random assortment. Those are some of the variations caused by gene recombinations, and they cannot result in they don't result in we call it in evolution. They are not very important for evolution compared to these other variations that are caused by mutations. Because when there's a mutation, then it means that that cannot be easily reversed, and then it can have a significant impact on evolution. Because, for instance, if an organism doesn't have the ability to acquires the ability for resistance to a particular pesticide, it means that in the presence of that pesticide, that is selection pressure is great because of the pesticide, that organism will be able to survive and then evolve and multiply that's okay. There will be more of them that are surviving. But these other variations that are about recombinations, in one generation, they can be there, in the next generation, they can be reversed. So they are not really very important for evolution. Then someone is asking, explain why linkage does not result into variation, hence evolution of new species. Well, when there is linkage, it means that there will be limited random assortment of those genes, okay? Because there's what we call crossover value. So the closer the genes are to each other on the chromosome, the harder they are to separate when we have, not random assortment, rather, when we have crossing over. So when we have crossing over, it means that these genes are less likely to be separated from each other. And when that means they'll be inherited together. And that means there will be a limited variety. And when there's limited variety, it means that there will be limited chances for the arising of new species. In that case, I hope that is clear, Stalconi. And then someone is saying, is it possible to have new species without variation? An interesting question. Well, I would say no, because new species, what makes a species different from the other is that it is having variation. Otherwise, if they're the same as the existing species, then then there's no variation, and therefore we can say they are different species. I hope that answers you. Someone claims they're not seeing anything on the screen. But my screen shows me that I am sharing. So I don't know, maybe there's a problem on your side. Someone was talking about significance of polyploid in plants. I think I talked about that. So let's get some answers for part A, part Roman 1. Do we have any volunteers? Read through the question and try to answer. With me talking all throughout is that I will rob you of the opportunity to exercise and learn a little more. Okay, it appears like people are very quiet. Okay, let's move on. So if we are going to look at, yes, Arina Itwe? Arina Itwe, please answer. Okay, Nitwe. Let's hear from the chat. In the chat, we can see for red, someone says, capital W, small w, capital R, small r. Okay, that one is fine. Okay, and then Our Lady of Fatima, capital W, capital W, capital R, capital R. That's okay. We see, so for red, all we need is to have all we need is to have at least one capital R and at least one capital W. Okay. Underline the word at least. Yes, Skyland High School. Seems some people are muted. I can barely hear. Okay, please unmute and talk. I don't know. I think there's some problem with their device. Okay, let's carry on. Keep the messages coming in the chat. Okay, so that is for red. Then what about for white? For white and then for yellow. How would you represent all the possible genotypes for white and for yellow? Maybe they have given us a hint. So for white, what do we have? And for yellow? So for white, somebody has put all of them as small letters for white. That is one of the possibilities. And then this is HP Light Book. Yeah, that is correct. Where I have small w, 2 of them, and capital R, 2 of them. So all we need for white to happen is that we have those W's, okay? When they are small, all of them. Doesn't matter whether red capital is there or red small is there, but as long as we have only W's. Then what about for yellow? So for yellow, all we need is to have at least one capital W, and then the R's must be recessive as it is written here, okay? So these are the answers I have. Then after that, we are taught to do a cross between red and yellow colors that should be able to give us this outcome, okay? That should be able to give us this outcome: 3 red, 3 yellow, and 2 white. So this is what I have. So the moment you are able to do these genotypes, then the rest of the things should be easy. Now, when you're showing genetic crosses, it's important that you write these words on this hand side, on the left-hand side. You always start with parents' phenotype, and then you explain what it is. This is like writing an essay. So you say that parents are onions with red color mating with onions with yellow color. Then you earn a mark. Then parents' genotype are, then now you interpret what red color is and what white color is. Because remember, there are many possibilities of red color and white color. But which of those possibilities is going to give us the ratio that the question wants? So that's now where you try to work it out on the side before you can transplant it on your paper. So you see that in our answers, we have yellow and white. We have white. So we need to do a cross that will result in some white. And for us to have white, we must have these homozygous recessive W's, okay? Homozygous recessive W's. And for us to have homozygous recessive W's, it means we must have a small w here and a small w here, okay? and we must have just one of them, okay? So when you notice that, then it's going to be easier for you to do the rest. So you notice that in this case, there are up to 4 different gametes that are because of this. But then here, we only have a maximum of 2 different gametes. So that's why our Punnett square doesn't have 16 boxes, but only has 80 result boxes here. So when we do the crossings, then we are able to get the 3 red, 3 yellow, and 2 white. So which is a total of 8 offspring. So that is how we go about this question. Okay. I hope we are together on that, because I want to switch to another question. Okay, Benjamin, that is okay. Because we said for red, we only need 1 capital R at least, okay, and 1 capital W at least, okay. Then for yellow, we need at least 1 capital W. That's what we said. And for white, all we need is small w's. So the moment you have understood that, then the rest becomes easy to do. So let's move on. Now I have another question. Now this is a graphical question. Graphical questions are typical of paper 2. And paper 2 usually challenges candidates because it requires a lot of analysis. And paper 2 prepares us for the real life, because in as scientists, we are supposed to be able to interpret information and use it to make decisions. So here we have an investigation on a healthy adult. So the person is healthy. That is very important. We are looking at their blood glucose levels when they have eaten a complex carbohydrate. So blood glucose level, and then we are looking at a complex carbohydrate, not a simple one, okay? It's not a monosaccharide, it's probably a polysaccharide, because those are the complex carbohydrates. So these are the things that should go through your head as you are reading through the question. It's very important you read through the question, then you go look at the questions, then you come back and read through the text again before you can answer. So you should read through we usually call this the literature, or the information that is given. So read through it, then look at the questions attached, then come back and read through. This time it means you are going to be more focused, because now you have an idea about what they're asking. Then now you attempt the question afterwards. Remember, there are no marks for hurrying. You'd rather take more time analyzing, then when you start writing, you will take less time. So, paper 2, section A is 40 marks. And like I told you earlier, each mark should be done in 1.5 minutes. So if it is 40 marks, you should spend 1 hour, not more than 1 hour, attempting that question. So it means that if you plan well, 1 hour is enough for you to answer the question. So don't panic, don't rush over it, but again, don't take too much time analyzing the question, because you will not have enough time to answer section B questions. So we are seeing that the experiment was repeated on another day, okay? Means this person had several days doing the same experiment. But this time the person ate protein, so the substrate has changed, okay? And on the 3rd day, the person ate fat, okay? So the person ate this food on separate days. And then they plotted the results when they were analyzing their blood for these different substances. So these are number of hours from the time the person has eaten the meal, and then they analyze the blood sugar levels. So when someone eats carbohydrates, so this is how the blood sugar level varies over the course of 6 hours. But when they eat proteins, this is how it also varies. And when they eat lipids, this is how it also varies. So we are going to study this graph and then use it to analyze the question, to answer the questions. So it's very important that you understand from what topic this question has been set. And then from there, you try to gather all the facts that you know about that topic. And then see which facts are relevant to answering this question. You don't put all the facts that you know on the question, because the question may be a bit restricted in the answers it needs, or it may be open for you to answer it with other points that you know. So my question to us is, what topic do you think this was taken from? Or what groups of topics would help us answer such a question? What groups of topics or what topic would help us answer such a question? Any ideas? Yes, Ruega Ahmed. Please unmute.
Osmoregulation and homeostasis. Osmoregulation and homeostasis.
Okay, homeostasis. Good. Skyland. Okay, he's put down the hand. Ruega, you want to tell us something more? Okay, he had forgotten the hand. Kangulumira, public access. Please unmute. Since people have a problem with their mics.
Yes, principles of homeostasis, osmoregulation and
Osmoregulation? Not quite.
the topic is
yes,
is homeostasis, excretion, osmoregulation.
Okay, that's the broader topic. But now when we go to that topic, we need to look at the exact areas. Okay, but homeostasis of what? What are we trying to control? They're trying to control blood sugar levels, what we call blood glucose. In the chart, I can see it's quite busy. Chemicals of life, maybe to some extent. Nutrition and homeostasis, true. Homeostasis. Chemicals of life, only to a limited extent. Respiration, yeah, a little bit of it. Osmoregulation, not really. Okay. Yeah, so you're getting it. So now let us look at the questions. Here you are asked to, I hope the graph is not too small. I decided to put it side by side with a question so that we can answer while we are looking at it. So we have, we are asked to compare. The keyword here is compare. To compare the level of blood glucose in the person after eating carbohydrates meal and after eating a protein. So are there any differences when someone eats protein or when they eat carbohydrates? When you look at this graph, do we see any differences? Yes or no? Do you see any differences? Do you see any similarities? Yes, help 24. Please unmute. Help 2024. Okay, they're having a problem. Let's ask Opwenye Joseph to unmute. Opwenye, we can't hear you.
Yeah, I see there are some differences.
Okay, that's a good start.
I said I can see some differences.
Yes, I didn't hear what else you said. Now, when you can't talk on the mic for one reason or another, the chat will be a better place because I'll still get to see your response. Katale Meka Mese Modern SS. Please unmute. Okay, in the interest of time, let me just give you some guidance. So if you are going to look at the differences after we've eaten the meal, you're going to compare similar time zones, because on the lower axis here, on the x-axis, we have time. So you look at the same time period, and then you make a comparison, okay? So if we look at protein, for instance, between 0 and 1.5 hours, notice that for the protein, they are both increasing, okay? That's a similarity, but then the increase isn't the same. So here we are going to now qualify or describe the extent of increase. So here we see that there is a gradual increase in the blood glucose concentration after eating protein between 0 and 1.5 hours. But then here we see that there is a rapid increase . One and a half to 1 hour, the blood glucose levels are decreasing, okay, while these ones are continuing to rise. So we are still maintaining the same area. Then from 1 hour to let's say 2.5 hours, the carbohydrates, when we take carbohydrates, the blood glucose level decreases while that of the proteins continues to increase. But now here it's important to, okay, here we may not qualify. You can just say decrease and this one increases. Since they are both not increasing or decreasing, you may write about the extent of decrease and the extent of increase, or you may leave it out. It's not such a big deal because they are clearly different. But putting rapid or gradual would put you in a better place to earn a mark, okay? Yes, Opwenye Joseph. Okay, I'm seeing Taiba saying carbohydrates increase rapidly, proteins increase gradually. It's a good observation, but you will not earn marks because you have not told us where is your reference point to write that comment. Is it at 2 hours to 3 hours, 3 hours to 4 hours? Is it the whole duration of the experiment? So that's why I always emphasize, first look on the x-axis. comment on what is there, and then make a reference to the y-axis. And when we're making a reference to the y-axis, we don't write figures. We only use descriptive words. Words like rapid increase, rapid decrease, gradual increase, gradual decrease, remains constant, acquires a peak, okay, acquires maximum, or acquires minimum. Those are the words that we use when we are describing what has happened on the x-axis. I can see Taiba is asking, is it right to say highest maximum peak and lowest minimum peak? You can't have a minimum peak. You have a maximum, okay? So the maximum, then we can have a peak. Now, you can have many peaks, okay? Like they can tell you that Mount Renzori has maybe 5 peaks. Mount Stanley, Mount maybe Whitney, Mount something like that. But the maximum is usually the highest peak. So when you're using the word maximum, make sure you're talking about the point that is the highest above all other points. Then if you have a number of high points, then those ones are simply peaks. Okay, so you can say the highest peak, then the others would be simply peaks. Then the lowest, if we say lowest, like for example, if you look at this, what I'm illustrating here with my red dot, so this is the lowest because there's no other point lower than this. So this becomes the lowest. And this becomes the highest because there's no other point higher than this. So this becomes the peak because it is the highest point. Okay, we could even have had another one around here as a peak, but then this would now become the maximum. So use the words peak and maximum carefully. They cannot be interchanged. You may interchange them and then they may cost you marks. Yeah, so I hope that has answered your question. So then we compare this other area. Notice that here, the carbohydrate is starting to rise again gradually. But what is happening in the same point on the proteins? The proteins are continuing to rise, okay? Again, still gradually. So it means that this and this are similar. This increase is gradual, while this increase is equally gradual. So that cannot be a difference. Rather, it would be a similarity from this spot to this spot, okay? Then we notice that from 4 hours to 6.5 hours, the blood glucose remains constant for when the person ate proteins, but rather carbohydrates, but when the person ate proteins, the blood glucose levels start decreasing, okay? And that decrease is rapid, okay? It is rapid. So that is something you can look at. So the point here is compare similar zones on the x-axis. Kamodo SS says that a carbohydrate acquires a peak before 1 hour, while the proteins acquires a peak at 4 hours. Okay. Now it is better you say carbohydrates acquire the peak at 0.5 hours. Okay, or 30 minutes. Tell us when that was acquired. Since you told us here it was acquired at 4 hours, so you also tell us this one was acquired at 4 hours. Okay, or you say it was acquired after 4 hours, then it was acquired after 30 minutes. So your expression is to be comparable. It needs to make sense. Okay. So those are the answers I have. So let's look at the ones I had prepared. So you can see blood glucose levels rises very rapidly immediately after the carbohydrate meal. Here it rises gradually after the protein meal. Then attained a higher peak. This one attained a lower peak. That's also one difference. You can go back. You see that the peak here is at 120, the peak is at 140. Then attained a peak earlier. That's what Kamodo was trying to say. This one attained a peak later, except that for him, he even told us that time, but it's the same answer. Then from 0 to a half hours, it increased rapidly. I think we talked about this. Then here it increased slowly or gradually, okay? Then from 1 hour to 2.5 hours, it decreased very rapidly, okay? Here it increased gradually. I prefer the word gradually rather than slowly, but if you write it, they will not deny you marks, but it's better to go with the better answer, okay? Go with the better answer. Then from 2:30 to 3:30, it increased slowly. Then here, from 2:30 to 3:30, it increased rapidly. So you can see these are clearly different from each other. Notice that blood sugar level, blood glucose levels decreased to a level below norm, and here it decreased to norm for protein. Now, this is where the homeostasis comes in. The normal blood sugar level for a healthy person is 90 milligrams per 100 cubic centimeters. Now, notice that here it has fallen below normal, what we call hypoglycemia, okay? Hypoglycemia. So it means that this person, blood sugar levels have gone below the normal. So that's when other homeostatic means set in to do a correction of this a deviation from the normal. Okay, so we see that now it rises up and goes back to normal. Okay, but proteins, for them, they rose above normal and then came back to normal without going below normal. So that is one of the differences. So I hope we all now know how to arrive at differences. Someone is wondering whether it's proper to mention what is happening at 0 hours when giving similarities. Well, that depends on the question. You see, the question said after the meal. So after the meal, it means that is from 0 going onwards. So the 0 hours in this particular question does not arise. And besides, at 0 hours, the blood sugar level was 90 for all of them, okay? Means we started with a healthy person, a person whose blood sugar level is normal. If we had an unhealthy person, then probably the blood sugar levels at the beginning may not have been 90, probably would have been more or far less than the 90. So for this particular question, we don't look at 0. But in certain questions, the 0 mark may be scored. So let us try to understand the principles of the questions. Let's not just cram, because one size doesn't necessarily fit all kinds of questions. Elite book says rapid increase and increase rapidly. Which one should be used? The two mean the same thing, okay? The two mean the same thing. So use whichever is easier for you, okay? Because I could say there was a rapid increase, or I can say it increased rapidly. We are meaning the same thing. So that should not be a bother to you. So we have another question. Here they're asking us to explain the observed blood sugar level, blood glucose level in the person after the carbohydrate meal, 0 hours to 2.5. So I put the graph here for us to be able to appreciate. what that change is. First of all, that is going to be scored, but then the reason behind the score, the observation, that's why the explain comes in. Explain means that you describe what you're seeing, but then give a reason for that description. So from 0 to 2.5 hours would place us somewhere around here. Okay, sorry, to place us right about here. So what is happening? Oh, 2.5, sorry, my bad. It's here. I thought 1.5, not this one. So it places us right here. So we are going to give a description for all this up to here. So when we look at this graph, you notice there are two distinct parts on the graph, the rising one and the decreasing one. So how do we account for this increase and then this decrease? And notice that this increase reaches a peak, so that is something to note. And this decrease reaches, this decrease reaches the lowest point here. So how do we account for them? How do we account for them? How can we account? Do we have, okay, I see the chart. The chart is a little busy. That's good. Do we mind the tense when answering? If it's an experiment, which happened, of course, in the past, then past tense should be better. Okay. Past tense should be better because this is something that happened in the past. But sometimes they are not too strict. However, I would prefer you do what is most appropriate, which is write in past tense. Is it okay to use very slowly in a description? No. It's either gradual decrease or rapid decrease. Because how slow is slow for it to be very slow? You see what the challenge? To some person, very slow may be gradual. You see, so rather than leaving us in the mercy of the examiner, it's important that you write things that are very clear. So KLC, don't write very slowly. Simply write gradually slow, gradually decrease or rapidly decrease. The topic of the question has been talked about in the chat. It is homeostasis, Nalango 16. I'm sorry about the noise in the background. Where I am, there are neighbors that I cannot stop. I'm so sorry about that. Can we have the answers? Yes, Napak. Napak, please answer.
There?
From 0 hours to half hour, the glucose level after eating a carbohydrate meal increase rapidly. This is because the saccharides of carbohydrate is a primary source of glucose. For example, starch. Upon hydrolyzing it, it causes the glucose level to increase. Then from half hour to 2.5 hours, the glucose level after eating carbohydrate meal decrease gradually. This is because the glucose level being at the higher level or beyond the norm cause the release of insulin hormone from the pancreas. So the glucose level was converted into fats, which was stored in adipose tissues, thus causing a decrease in the glucose level.
Okay, thank you, Napak. Thank you, Napak. Okay, I think let's try to save the time. Let me share what I have. So in line with what Napak has said, the level of blood sugar, blood glucose increased rapidly. Actually, this one very should go off. So I want to remove increase rapidly because after eating the carbohydrate, it was hydrolyzed. You know that starch is usually hydrolyzed into glucose, first of all from maltose, then to glucose, then which can now be absorbed into the bloodstream. So that means now the blood sugar will shoot up. Then it increased above the norm, like Napak said, it's far beyond what it should be, which caused the beta cells of the islets of Langerhans in the pancreas to secrete insulin. So these blue words are just the explanations for the observations. The words in red are what we observe on the graph. I wanted to spell that out very quickly. So when insulin is secreted, we need to talk about where. Whenever I talk about anything being produced in the body, it's important to talk about where it is produced and where its effects are, okay, and why it's produced. So here we see what has been produced is insulin. It's produced by the islets of Langerhans, and its effect is on the liver, okay? So the liver is able now to convert the excess glucose. You must add the word excess, not just glucose, but excess glucose to glycogen. And some of the excess glucose may be respired, or it may be used to form non-carbohydrates. What Napak was talking about, the fats, the non-carbohydrates which are stored away, okay? So when all this we have said has happened, then we see that the blood glucose levels will rapidly decrease because the rate at which they are being converted into non-glucose, non-carbohydrate, and also respired and converted into glycogen is very high. So that's how we would go about such a question. I hope that is now clear. Then we look at the next question. Mugwanya, let me see. Mugwanya has written something that from 2.5 hours, the glucose levels were lower than normal. Okay, he's attempting the second question. This was detected by the alpha cells of the islets of Langerhans of the pancreas secrete glucagon hormone, which aids in increasing glucose levels back to normal by causing conversion of stored glycogen liver and the muscles to glucose, reduce respiration rate. Yeah, that is okay. Someone says, but I was told to use gradually and rapidly only in comparing changes in the graph. That is true. That's why this was a mistake, and I have clearly stated it. The vary and the The very gradually and very rapidly are not correct. So just simply use rapidly or gradually, KLC. So that was a mistake, and I have clearly spelled it out. So in line with what Mugwanya has said, that is the answer. Okay, we have something in the chat. Someone is saying negative feedback since it's below the norm. It's correct, but we are more interested in what is involved in that negative feedback. Okay? But I would rather you don't talk about negative feedback. You just simply tell us what is involved that is bring back glucose levels to normal. Okay? Other than saying negative feedback. Okay. Let us move on to the next question. Okay. So here they're asking the blood glucose levels in the person after the protein meal. So, and also lipid meal. We know that proteins are not carbohydrate, neither are lipids, but yet they are causing a change in the blood sugar levels. How is that possible? Yet they are not carbohydrate in nature. How would they influence the blood sugar levels? Someone says I'm too fast. I'm sorry if I'm too fast, but I'm trying to move slowly. I had prepared 3 items, but it appears like I'm going to only discuss 2 of the items based on the time we have. I was hoping that we may do all the 3. Someone is wondering whether on comparisons, we should not include similarities. Comparisons can also include similarities. And I talked about the similarities when I was discussing, just that I did not include them on the presentation. Okay, so let's continue. So why is there a change in blood glucose levels even when we take proteins and lipids? That's the question. So I'm not seeing any answers in the chat. So we see that from 0 to 4 hours, the blood glucose level first increases slowly. You see that? For proteins, okay? Then rapidly towards the peak. Let me see. Will you say that? I think no, would rather say, let's see, this gradual then, okay. There's a certain change in the tangent, although generally speaking, this is more or less the same. Okay, so it increased gradually, okay, towards the peak would be more like the answer. Because if this tangent had gone on like this, let me just draw it. If it had gone like this and maintained like that, then that's when we would say it increased rapidly. But because when we look at it, the tangent, it hasn't changed that much. Because if I was to extrapolate like this, I'd still arrive at more or less the same point. You see that? So this would probably just be, it increased gradually. Because again, with biology, if you cannot account for this anomaly, then you're better off not saying anything about it, because it will cost you some, it will be difficult for you to explain it. So if you can't explain it, then don't mention it. Okay. Then we are saying proteins are first hydrolyzed to amino acids, okay, during digestion, and then absorbed in the bloodstream. Then remember, when we eat excess protein, then we have excess amino acids, which are then deaminated. So the deamination means you're going to remove the amino group and then use it to form ammonia, which will be excreted as urea. But then the remaining part of our amino acid, which is the ketone, is converted to glucose slowly. So that is why you see that the rise is gradual. Okay. If it was being done rapidly, then the rise would also be very rapid. Okay. Then from 4 to 6.5, notice it decreased rapidly. So it means that blood glucose that was present is now being respired. So it means that it's going to decrease. And because the rate of respiration is high, the rate of decrease is also equally going to be very rapid. So that is why we see that. Then for the lipids, we notice there's also a gradual increase. Okay. There's a gradual increase to the 3rd hour, and then a gradual decrease afterwards. Okay. So why is there a gradual increase from 0 to 3 hours? We see that lipids are hydrolyzed to fatty acids and glycerol, which is generally a slow process. And then these are later on converted into glucose. If you remember the Krebs, no, not Krebs cycle. There's a diagram in your textbooks that shows how other sources of how other substrates can be hydrolyzed to generate energy by the body. So if you look at that diagram that shows how proteins are involved in generating energy, how lipids are involved, and how carbohydrates, you will notice the role of fatty acids and glycerol in generating energy. And because these two are put in the fatty acids join in glycolysis, okay, and glycerol, so they have to go through all those stages before now the energy can be released through Krebs cycle and through the hydrolysis of the NADs and the fats that are generated by those processes. So these processes are very slow, and that's why we see that the blood glucose levels take long to reach their maximum, and even when the maximum is attained, it is not that high compared to other substrates. Okay, so that is very important. Yeah, but the good thing about it also is that the rate at which it decreases is very gradual compared to any of these. So it means when you eat a lipid, you take longer to feel hungry because the rate at which the glucose that it has generated is utilized by the body is rather slow. Okay. Someone is wondering the meaning of deaminated. The word D, when you see the word D, it means without. So amino and aminated comes from the word amino. So if you look at an amino acid, it has an NH2 group. Okay, let me try to write an amino acid here. Where's my notation? Okay, here we go. So just give me a moment. So if you recall that an amino acid has N, which it has H2, N, then you have the C, then you have the H, and then the R, then you have the C, then OOH. So when we look at the amino acid, it has this bit, what we call the amino group. Then it has the carboxylic group. So when you eat excess proteins and they are hydrolyzed into amino acids, these amino acids that are in excess must be deaminated because excess amino acids are not stored by the body. So to deaminate means to now get rid of this amino group, you see? So what remains what we call the carbon skeleton, okay, or the residue. So this thing that remains is what can be funneled in those in the Krebs cycle at different instances, and then they can generate energy for the organism. So the amino group later on forms ammonia, and then the ammonia combines with carbon dioxide to form urea, which is excreted out of the body. So that's what we mean by deamination. So deamination is a removal of the amino group from an amino acid, okay, and it's done by the liver in order to get rid of the excess amino acids. So whatever remains can be used to generate fats or to form even other carbohydrates that are stored by the body. I hope that answers you. The one who asked earlier. So St. John's Kabu, the carbon skeleton is what remains when we remove the amino group. Some books call it the residue. Okay, the carbon skeleton, others call it the residue or something like that. But basically, it's what remains when the amino group has gone off. I hope that is clear. So I'm encouraging you that after this meeting, you go and revise the areas where you find that you were not at par with discussion, so that you do not miss out. Okay, let's go through this quickly, then we shall call it a day. So explain the difference in variation of glucose levels after carbohydrates and those of protein. So why do we see a difference in carbohydrate level of glucose levels after eating carbohydrates with that after eating a protein? What is that? What causes that difference? Okay. So the difference here is that blood glucose levels after taking the carbohydrate had a higher peak than after taking a protein. So you can see this right here and this right here. That is the difference. Okay. Notice the question wants that difference, not differences. So then, because they want us to explain, then we must give a reason why it is as such. So that reason is that carbohydrates, when they are hydrolyzed, we form glucose right away. But for proteins, we must first of all form amino acids, then they must be deaminated in order to form the glucose. So that whole process takes time, okay, and it is slower. Then the significance of the physiological process illustrated here. So here we see that glucose is actually the main respiratory substrate, and it is mostly used by cells such as the brain cells. For them, they will not use any other substrate. And you will notice that when you are thinking actively or when you're in the season of exams, you get hungry faster because your brain cells use a lot of glucose, because that's the only substrate that they can respire. Then we also notice that lack of glucose in the body will cause hypoglycemia, which will result in fainting. Okay, you've seen people fainting when they have not eaten It's because of hypoglycemia. And remember, glucose is the fuel of your body. Without glucose, your body will not be able to do most of its energy demanding processes. Then it will cause ketosis. Ketosis, if you notice when people are fasting, they usually have a certain breath. Their breath smells in a certain way because of those ketone bodies that are being broken down, okay, during fasting. Yes, Skyland High School. Then we also see that the glucose concentration would affect osmotic relations of the cells, because you know that glucose attracts water, okay? So if we had excess glucose, okay, then it would mean the brain cells will take in too much, and then as a result, taking much water, which would cause them to undergo hemolysis, which will cause them to break down. So excess glucose is equally not good, okay? Yes, equally not good. So that is the other thing we notice from this. Yeah, so the seminar, someone is wondering what time it will end. The seminar is about to end. Take note, okay? Then ketosis, you'll go and read biological science in the topic of nutrition. There's a place where they talk about the different substrates. There's a graph which shows you someone who is starving, and then they show you the rate of breakdown of those different parts of their body, glucose, the carbohydrate, the lipids, and the fat, and the proteins. There's somewhere where they make a reference to the word ketosis, but ketosis is basically noted when people are fasting. They tend to, their breath tends to smell in a funny way because of the biochemical reactions that are actually taking place when they are breaking down other sources of substrates for their respiration. Okay, I guess that will be it for today. The other question I had, time has not allowed us to go over it, but was something concerning the countercurrent multiplier and the countercurrent heat exchange system, something like this. So that will be it. So that will be your homework. You go and find the differences between the countercurrent multiplier and the countercurrent heat exchange system, its importance and how they both operate. Maybe a quick one, which I'll not let go, is why are people given a salt and sugar solution when they have had diarrhea or they have vomited excessively? Why are they given ORS, which contains sugar and salt? So that will be our closing question. Someone is wondering whether this can be downloaded on YouTube. I believe they are available on YouTube. The help organization usually uploads all these things that we do. So you can talk to Mr. Kaziba, then he'll give you guidance on how to access this information. That goes for everyone if you can access YouTube. Okay. So the last question I said, why is someone given a sugar and salt solution when they are dehydrated after vomiting or having diarrhea? Why don't we just have only water? Or why don't we just have only salt? Or why don't we just have only sugar? Why do we mix the three? Or why do we mix sugar and salt? Why is that so? Can we try that out? Just one person, then I'll display the answer. Okay, people are leaving the answer. So here is the answer. You can screenshot it and go and think it over. So, but it's basically to do with the fact that sugar and the salt are co-transported. You know, if we talk about co-transporting, we have antiport, symport, and the uniport. Antiport means that two substances are moving in opposite directions across the cell membrane. Uniport means only one substance is moving. Then symport means two substances are moving in the same direction during absorption. So during absorption of glucose, even salts are taken up. Okay, so it's important to combine the two, because wherever salt goes, even water will follow. So when the salts are absorbed into the system, then water will equally be reabsorbed. Okay, but then secondly, it helps to restore the electrolyte balance, because when you diarrhea or you vomit, the amount of salts in your body reduce significantly, and therefore the electrolytes in your body will reduce so much. So by taking ORS, these electrolytes are going to be restored. Yeah, so that is where the answers lie. For your time, I hope this inspires you to work harder. I wish you the best in your preparations for mock. God bless you.
Yes, thank you so much, Teacher Crescent, for taking us through the biology facilitation. We are really very grateful, and we can't thank you enough. Thank you so much for the efforts, for the wonderful presentation. We want also to thank all the teachers that have been on this call and all the students from the different schools. Thank you so much for joining, and thank you so much for putting in your time, the data, and everything. The presentation is going to be shared. It's going to be on the YouTube, and you can be able to access it at your convenience anytime you want. Starting today, you check from 9 p.m. onwards, it will be there. Just type in the Holistic Learning Platform YouTube, and then you can be able to access even any other seminar that you have missed. You can be able to find it on that platform. Everything you need, we shall provide, and we shall do all what it takes to ensure that you are able to do your exams successful and that you can be able to pass highly. Tomorrow we shall have another session for another subject. Kindly keep time for tomorrow's program. We shall have 2 rooms, but for your friends at school that do geography, kindly let them know that we shall use a different room for tomorrow for geography. This one we shall leave it, we are going to leave this room for the other subject, but for particularly geography will be in another room such that we don't mix up the recordings. But in case of anything, kindly, we shall be posting in the WhatsApp group where your teachers are, and they'll be able to let you know. Once, thank you so much, and let me wish you a good evening. Bye-bye, everyone.